Today we're going to start a new series whereby we analyze the images associated with the various laboratories in the histology class. This particular one has to do with images on fundamental tissues and mitosis. It has three parts. One part talked about the types of microscopy, tissue preparation, and staining. The second part has to do with images of light and electron microscopy, and the third part has to do with mitosis itself. If these materials are useful to you, I hope you'll be able to pass this information on to your colleagues and to your friends to have them subscribe to the YouTube site. Thank you.
Hi, this is Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about the images associated with the fundamental tissues and mitosis, which is the first medical school laboratory we have in histology. To begin with, we look at protoplasm as living substance. Living substance, and a single unit of protoplasm is a cell. A cell is, some animals only have a single cell. Groups of cells with the same general function is a tissue. You got muscle, nerve, epithelium, and connective tissue. All those are tissues, which are groups of cells doing the same thing. Also, there's organs. So if you go from tissue to organs, two or more tissues with a similar function is also an organ. And then organ systems are several organs working together in systems. Cells, tissues, organs, and organ systems is the way the body is made up. Protoplasm, smallest unit is a cell, and that is the smallest unit of living substance. And if you look at a cell composed of a nucleus in the center, and then a cytoplasm around the outside. And in that cytoplasm, we may have a Golgi apparatus, we may have rough endoplasmic reticulum, which are sets of membranes with ribosomes on the surface, nuclear pores, lipid droplets, mitochondria that would be in through there, which is a double membrane structure. Smooth endoplasmic reticulum, sets of tubes there with no ribosomes on the surface, centrioles, or it could be secretory granules. So all of these guys, all of these structures here are part of cytoplasm, and here is the nucleus. So this is a cell. If you have groups of cells with similar function, that makes a tissue. More than one tissue makes an organ, and more than one organ makes an organ system, and that makes the individual. Now if you look at the interphase nucleus, here's the nucleus. You see euchromatin and heterochromatin around the edge, as we see the darker part of the DNA material that's inside there, DNA and protein, of course. We have nuclear pores and several. We have intermediate filaments and microfilaments, microtubules that radiate from a centriole. So this is the nucleus, interphase nucleus. And if we look at these different cells, here's one cell here, a pyramid type cell. You see a euchromatin, lighter chromatin in through this cell, and there's secretory granules in this particular cell. This is the base and this is the apex. This is a cardiac muscle cell, and you see the cytoplasm in through here. Most of these are the myofibrils that we're seeing here, little cross sections of the actin and myosin components in through there. That's the pink. And then we see the nucleus. There's a nucleus here, a darker nucleus in these cells, and you can see a darker nucleus in blood too. This is a white blood cell. There's a neutrophil, eosinophil, and there's a lymphocyte. So different cells. So the cytoplasm of this lymphocyte is a little bit right there. There's a little bit right there, but the nucleus is a dark structure. In these intestinal absorptive cells, we see this is a single cell here. So it's a simple columnar cell, simple one layer columnar. And note the brush border. These are projections off of the cell, microvilli of uniform length and high density as well. And so here we see the nucleus. It's got a euchromatin, heterochromatin that we see there. You can actually see mitochondria too very nice mitochondria that we can see there in the cytoplasm. Nucleus, cytoplasm. Here is a blood vessel, or white, that's a red blood cell, and this is the endothelial cell. You can see the endothelial cell nucleus there. There's another endothelial cell. Can't see the nucleus thereof. Another endothelial cell, another endothelial cell. And so epithelial versus endothelial, and these are lymphocytes. And this is the nucleus with the heterochromatin on the outside and the euchromatin on the inside. These are goblet cells in through here. So these are the nuclei of the intestinal absorptive cell, and here are the cytoplasms of these different cells, which have the various components that we just saw a while ago. Now, in routine staining, we usually use hematoxin and eosin. Hematoxin binds a nucleic acid. Nucleic acids are dark staining. So something with a high acid content will stain hematoxin. In contrast, the cytoplasm is proteinaceous. Proteins stain an acid dye, eosin. So here we can see these are smooth muscle cells, and we see the nuclei, which are little dark things, but we also a lot of pink. It's the and so we're going to see those cells. First of all, those were lymphocytes that we saw of tonsils. Here's stratified squamous epithelium in through there of the opening into the oral cavity. And here we see the germinal centers. So these would be where the lymphocytes are that we'll be looking at in our specimen. So if we take a look at that, so when we look at the tonsils as one of the ones that we're going to see, we see a germinal center. So these are different germinal centers, and there are going to be lymphocytes located in there that we'll be looking at. This is stratified squamous epithelium associated with the oral cavity. This is a tonsil, so this is the tonsils in the oral cavity. So we'll be looking at lymphocytes located in there. And so if we look at this, this is the image that we will be looking at. We can see the germinal center. This is a germinal center, and we know this is stratified squamous epithelium here. And so if you have lymphoidal tissue associated with stratified squamous epithelium, you got to be in the oral cavity likely. You could have some in the trachea too, but it's mostly going to be, especially this high density, is going to be tonsils. So if we look at this, we should be able to see here, we see some metaphase plates of cell division, but these are all lymphocytes in through here. And these lymphocytes have a very dark nucleus. They're staining with hematoxin. Now, in contrast, there are some other cells. This is a smooth muscle cell here, and you see that its nucleus from there to there, with a nucleolus in the center, is more euchromatic. So heterochromatin with a dark nucleus, or euchromatic with the lighter nucleus. Another one that we're going to look at is the smooth muscle cells that are in the transition between the esophagus and the stomach. So we had stratified squamous, more than one layer of cells here, and simple columnar cells in through there. We can see there's blood vessels in here. We can see the endothelial cells lining the blood vessels. This is a simple columnar epithelium here, stratified squamous, more than one layer with flattened cells on the surface. So that's what we're going to see here. Here's the slide, and we can see the stratified squamous epithelium coming down through here. That is, there's a basal layer of nuclei, and then more cells all the way through. Basal layer of cells come all the way through non-keratinized stratified squamous epithelium. That is the esophagus, and it stops here very abruptly, and we pick up these columnar cells, simple columnar cells here. This is the nucleus of these cells that we can see. But what we want to see is the muscle cells. This is the muscle that we can see. The muscle cells is very pink. You can see the nucleus in through there, but the cytoplasm is very eosinophilic. So this is the smooth muscle cells that we see in through here. We can see the kind of long shape of the smooth muscle cells. This is an endothelial cell in through here. There's 1, 2, 3 of them. White blood cells, red blood cells, and you can see the swirls here of the smooth muscle. Longitudinal view of the smooth muscle and cross sections of these smooth muscle cells. And so we can see that. Here we can see a longitudinal view of the smooth muscle cell. It's like shaped like a cigar. Here's the nucleus, and this is a cytoplasm. If you cut it in cross section, it would look like this with this nucleus in the center. The next one that we're going to look at is the eosinophilic staining of the proximal tubule in the kidney. So this is the kidney. Here's a capsule in through there, capsulated organ. This is the cortex and the medulla. And so we're looking right in through there, and in there you have the nephron. The nephron, of which starts out in through in the cortex and goes down into the medulla. This portion here is in the medulla, a thin loop of Henle, and the collecting duct. The collecting duct takes urine out through here. So the blood interacts with the nephron, this being the nephron, and it filters the blood. So you got the fluid in through here, and then absorption occurs, or secretion occurs, and then ultimately you end up with urine, and 99% of the water is being returned. So what we will see, again, we're in the cortex and in the medulla, and here we can see the proximal tubules and the very eosinophilic staining thereof. In contrast, this is a distal tubule. It's not as eosinophilic a distal tubule. Also, in the proximal tubule, nuclei are far apart, but in the distal tubule, nuclei are close together. Also, if you look in the proximal tubules, you see this flocculent material inside there. This is the brush border. There's a brush border, uniform microvilli, a high density in the proximal tubule, but not in the distal tubule. And so, if we take a look at that specimen, we can see the cortex in through there, cortex from here to here, and then the medulla all the way there, and then finally urine is released into the pelvis and through there. And so if we look at a higher mag of the cortex, we can see the capsule surrounding the kidney, and then we can see a lot of tubules inside there. And these in through here, but very eosinophilic staining, which is what we're supposed to look at. So this is pink staining, it's the cytoplasm, and the dark blue staining is the nucleus. And inside the nucleus, you can see a little dot, that's the nucleolus. So these are the proximal convoluted tubules, and these are the distal tubules in through here. And there's red blood cells going in through there that maybe should not be there, but they're there. We can see a little capillary. You can see red blood cells here that are in the capillaries that are in between there. We were the other thing that we were supposed to see is another staining is periodic acid shift, which stains for carbohydrates. It stains for carbohydrates, and we can see that the base of epithelial cells, this being the base and this being the apex, is stained. The basement membrane is stained with PAS staining, indicating that it's carbohydrate rich. In addition to that, the brush border, which remember is a high density of microvilli projecting off the surface of these columnar these cells. These happen to be cuboidal cells, but nevertheless off the surface of these cells, and that the glycocalyx is a very elaborate glycocalyx in this location. Glycocalyx goes all the way around the cell, but it's amplified right around the brush border, and that's why you see that pink there. And so if we take a look at that, we can see this is another kidney, and this is the cortex through here, and the upper part of the medulla. We don't see where the urine empties into the renal pelvis in this case. So if we look again, we can see just what we saw before, and in the cortex you will be able to see glomeruli. So we see this is a renal corpuscle, and these are capillaries in through there. And then here is a proximal convoluted tubule. You see a distinct PAS staining around it at the base, and also you see the flocculent brush border on the inside of these. In contrast, this distal tubule does not have this pink inside. It does have the staining of a base, because all epithelium sits on the basement membrane. So that's what we see. This is the glomerulus. That's a parietal epithelium. This is Bowman's space, and through that we'll learn about. So these are proximal tubules with the PAS staining brush border that we will see. That concludes that presentation.